Hello, and welcome to the Cell Press webinar on the visualization and analysis of intracellular dynamics. I'm Allison Evans, scientific editor of Molecular Cell, here with my colleague Gary Karpen, scientific editor of Cell.
The ability to track labeled macromolecules in living cells has taken cell biology beyond static images of fixed cells to a real-time view of cellular dynamics. Ever-improving technologies for molecular labeling, image acquisition, and image analysis have provided exciting glimpses of the trajectories of proteins, chromatin, and organelles during various cellular responses.
Today, we'll hear how intracellular imaging approaches have been leveraged in several different ways to teach us something new about how a cellular process works. First, Tobias Meyer of Stanford University will tell us about imaging techniques to monitor protein translocation in response to calcium signals. Next, Gary Karpen of UC Berkeley and the Lawrence Berkeley National Lab will talk about how damaged DNA within repeat rate regions translocates outside of heterochromatin to get repaired. Finally, Jennifer Lippincott-Schwartz of the NIH will give us a tour, so to speak, of how photoactivatable proteins and super-resolution imaging have yielded insights into the dynamics of organelles and the cytoskeleton.
Before we get started with the presentations, we'll go over a few practical points. Each of our speakers will talk for about 15 minutes. Then, we'll have a question and answer session that covers all three presentations. If you have a question for any of the speakers, or for all three, just click on the question tab at the top of the player and type it in. You can enter a question at any time. There's no need to wait until the end of the presentation. Allison and I will present as many of your questions as we can during the Q&A session.
And you'll see there are three other tabs on the viewer. One lets you provide feedback on the webinar. Another has speaker bios. And the third one you can click for support if you have any technical difficulties during the presentation. Now let's hand things over to our first presenter, Dr. Tobias Meyer. Tobias?
Yeah, thank you very much, Karen and Allison, for organizing this webinar. I think this sounds like a very exciting event. So I would like first, in my 15 minutes, talk about how imaging can be used to understand cell signaling processes. Particularly, I will focus on signaling at the plasma membrane, which is arguably the most important place where many signaling processes take place. So I will be focusing on calcium signaling in most of the points I'm making, but I want to also make some general points along my talk about how imaging can help us understand signaling processes in complex systems. So first I want to maybe mention that many people now believe that live cell fluorescence imaging of signaling events is the most powerful approach to dissect complex signaling processes that occur in real cells, where often many pathways interact with each other, and without really seeing signaling happen in single cells, it is very difficult to deduce that kind of information from more bulk analysis and biochemical studies. So there are currently two main ways to visualize signaling events in cells. One of them is based on fluorescence resonance energy transfer, and I will not talk much about this in this presentation. And the second one, I think, which has been useful up to date in many more contexts, is the translocation and localization and co-localization of signaling proteins in cells, which often was very useful in figuring out how activation steps work and how they function. And I want to start by talking about an example that got our laboratory interested in cell signaling, and this was many years ago when we were interested in understanding the function and activation of protein kinase C in cells. So as you see on this slide, we have been performing initially imaging of calcium signals in individual cells. So you see on the top these blue spots are each a cell with a calcium indicator in them, and if this indicator later when we play the movie will turn red, you will know that calcium went high in that cell. In the lower part of that image, you see the expressed green fluorescent protein tagged protein kinase C, which, as you will see, translocates from this cytosolic non-nuclear localization to the plasma membrane in response to an increase in calcium. When we first saw this was a very striking result because, as you will see, this translocation happens in only 2 seconds, and it made us think about the ways of how signaling proteins can move around in cells. And photobleaching experiments then showed that the diffusion of this protein kinase C is faster than 10 square micrometer per second, allowing it to passively, just by Brownian motion, to bounce into the plasma membrane several times per second. So when calcium goes high, what you will see is basically this random interaction with the plasma membrane gets stabilized. So if you could maybe play the first movie. Yeah, so as you see now, when you focus on some of these cells, the moment one of these cells turns up, it will translocate the PKC to the plasma membrane. Then when it turns dark, several of them start oscillating because calcium goes up and down during this time. And the localization of protein kinase C at the plasma membrane, where it functions as an enzyme, will follow the calcium signals. So maybe you could go back to the same slide. And in a number of site-directed mutagenesis studies, deleting and using isolated parts of the protein, we could use this imaging technology to show how protein kinase C is activated in cells. So first we found the C2 domains that bind calcium and phospholipids are used to cause that initial translocation to the plasma membrane. And then only in a second step, the diacylglycerol, which is also co-produced by receptor stimuli, will engage another type of domain in protein kinase C, C1 domains, and pull it to the plasma membrane. This model is shown schematically from a paper many years ago in the bottom part of the slide. So I use this as kind of a first intro into the power of using imaging technology to deduce something about the function of proteins. And I just want to show in a second movie that you will see on the next slide is from a study in glial cells where we found that that protein kinase C isoforms in a very similar way as you saw before, moving in oscillations to the plasma membrane in these tumor mast cells that we showed in the movie before. In this case, this is a sheet of glial cells that you would have in the brain, interconnecting neurons and transmitting information in that way. When we monitored with another technology called total internal reflection microscopy, the localization of this protein, we could see wave-type patterns by which these protein kinases translocate to the plasma membrane. Maybe if you could show these waves. So what you will be seeing is waves of protein kinase C translocation to the plasma membrane in response to glutamate stimulation. So glutamate is an excitatory neurotransmitter that causes these waves that are propagating information from one place in the brain to another between these cells. And these waves actually propagate across gap junctions from one cell to the next and cause a kind of wave-like activation of protein kinase C across a sheet of cells. So these are kind of the two approaches that are useful for studying plasma membrane translocation. One of them is just kind of confocal imaging, and the second one is total internal reflection microscopy, which both are powerful ways to visualize movement between the plasma membrane and internal sites of cells. So these type of, could you maybe move back to the slide? These type of approaches we considered then would be useful if we could design minimal domains instead of using full-length proteins. So the idea was that we could create biosensors that would selectively just monitor diacylglycerol and not also calcium at the same time. So instead of taking full-length proteins, we then thought it might be very powerful to cut out pieces of proteins that have very specific functions. So in a number of papers, we then try to do that for SH2 domains, for the C1 domain, as showed before, as part of protein kinase C, but also for other domains like the phospholipase C delta pH domain, which we first showed can be used in cells to monitor the concentration of plasma membrane PI45P2 lipids. We also found that another pH domain from AKT can equally be used to monitor the activation of PI3 kinase and the production of PIP3 lipids. This domain is now often used in studies of cell migration and chemotaxis and other types of projects. So what I wanted to emphasize here is that relatively simple tools can be generated based on existing protein domains and proteins in cells that tell us a lot about what and where in cells signals are generated. So these biosensors are in many cases easier to interpret and understand than FRET signals, which have additional complications. So I just want to introduce one method I think that is very powerful to study the specificity and function of some of these biosensors, which you can see here is that we found that if we pull many types of signaling regulatory proteins from the cytoplasm to the plasma membrane, for example, here in the movie we'll be running later, you will see the PIP2 phosphatase being pulled to the plasma membrane and hydrolyzing PIP2 to make PIP out of it. And on the right side of this movie, you see a pH domain from PLCT that monitors PIP2, and this will fall off the membrane as the movie starts running when we use this heterodimerization system based on rapamycin that was a long time ago developed by the Schreiber and Crabtree laboratories. So as you see here is that when this pulls to the plasma membrane, you see this instant dissociation of the PIP2 biosensor for the plasma membrane, proving that hydrolysis of PIP2 is sufficient to cause the dissociation of this. This kind of proves the specificity of this indicator in this context. And we have used that for many different proteins to study activation of small GTPases and other systems in cells. And I have a few references here on that. So what I wanted to do in the last few slides is to tell you an example of where we were able to use a similar intracellular translocation to figure out very quickly what the function is of a protein we identified in a small interference RNAi screen in calcium signaling. And I want to quickly explain that first. So there was a long-standing question in the calcium field is what happens after the activation of receptor and phospholipase C releases calcium from the endoplasmic reticulum, which was known to then be leading to an influx, an activated influx of calcium from the outside the cell into the cell. So the store-operated calcium influx or crack influx was known for 20 years without a known molecular player mediating that. So in the RNAi screen, we identified a protein that was called STIM, and I want to show how we figured out the function of that in a series of experiments. Just to take this away, initially, so there were two groups actually that discovered this STIM protein in parallel. It was our group in Stanford and also in California, a group at a company that was doing a Drosophila screen, and we did a human screen for these store-operated calcium influx sensors, and we found the STIM protein. And I should also mention in subsequent studies, 3 laboratories used RNAi screens and identified the protein ORI that turned out to be the calcium channel. But I want to focus on what I think is one of the more interesting regulatory proteins that I have encountered yet in cells, is this STIM protein, and how measuring movement in cells immediately told us how it might work in cells. So what we found is that when we express YFP tagged form of this STIM1 protein, that when we knocked that down, it eliminated the store-operated calcium influx, and when we expressed it, it was co-localized with the ER, and upon stimulation by fabsigargin, which releases calcium in an artificial way from the ER or by using receptor stimuli, we could see these puncta form near the plasma membrane. So this puncta formation was, we believed, a mechanism for that would help us understand what is going on here. So So in this movie here, I show you how dynamic this is, and this really led to please play the movie. This led us to understand that this is a reversible localization, where initially we thought the STIM protein might fuse with the plasma membrane, but since it was just coming back from these junction-like sites into the ER, we concluded that there must exist these sites in cells where the ER is very close to the plasma membrane, and we can reversibly move proteins from the ER into these ER plasma membrane junctions and back in response to reducing ER calcium level. And the next slide, please. Yeah. And in a number of studies, we were then able to show that in this endoplasmic reticulum there is a EF hand type domain that binds calcium, and we could make a site-directed mutant of that site, of that calcium binding site, and found that the protein then already pre-localizes to the ER and constitutively activates calcium influx into cells, arguing that it's indeed the sensor and mediator of this process. Similarly, we could show that these junctions are very near the plasma membrane using, again, TIRF imaging, which showed that actually there are sites of ER very close to the plasma membrane. And these sites are the locations where STIM translocates to and from, arguing really there is kind of predefined spots where the ER connects with the plasma membrane and STIM moves in and out in this configuration. And so this led to this junction hypothesis that STIM activates store-operated calcium influx by reversible translocation along ER membranes to proximal plasma membrane sites. And we could also show that translocation and activation is triggered by calcium dissociation from its luminal EF hand. And the main parts of these studies was based on site-directed mutagenesis and imaging type approaches that really solidified this hypothesis, and I think this would have been impossible to do without live cell imaging. Just as a note, so we were able to also show that this signaling is mediated by oligomerization of STIM, and I cut it a bit short here because I'm running out of time. And we also found that this STIM protein is cooperatively activated by ER calcium, explaining how a relatively small drop in ER calcium can lead to a very dramatic translocation and activation of calcium influx, and we think this is caused by this oligomerization. And also the two STIM proteins actually have a bit different affinities, so depending on the cell type, they can regulate calcium in a different range of ER levels. So just to finish up, so this led then basically to the model that calcium dissociation from a calcium binding site in the luminal side of STIM leads to oligomerization, that this oligomerization then creates an entity that can bind to the plasma membrane. We could actually show that this is likely PIP lipids because it involves the polybasic region, and this then causes the recruitment and activation of ORI, these calcium channels that then let calcium into cells. So just to summarize this, so I think what I wanted to point out is that in complicated cell signaling processes, that imaging using TIRF and confocal and other types of imaging approaches is absolutely essential to really get down to molecular mechanism and understanding how signaling pathways interact, from where to where signal information flows, and how all these converging and parallel processes are regulated in cells. So that was my presentation, and I want to turn it over now to Jennifer, who will have the next I think to Gary, who will have the next talk. Thank you.
Thanks, Tobias. Now we're going to move on to Dr. Gary Karpen and his work on the dynamics of repairing heterochromatin. Gary?
Yep, I'm here. So if we can go to the first slide, I'm going to tell you how live imaging has helped us to identify novel dynamic behaviors involved in DNA repair, and the title is Heterochromatin Proteins Regulate the Spatial and Temporal Dynamics of Recombinational Repair Among Repeated DNAs. And the significance of the solar flare at the bottom hopefully will become clear by the end of the talk. So first, let me give you a little introduction to heterochromatin, and I'm not sure if the next slide, let's see, is coming up. There we go. So about 30% of the fly in human genomes are considered heterochromatic, and what distinguishes these regions is that they're enriched for repeated sequences, including simple sequences like AATAT for hundreds of kilobases to megabases. Also enriched for what are known as silent epigenetic marks in proteins such as H3K9 dimethyl and trimethyl, and also heterochromatin protein 1 or HP1A. For the purpose of this talk, it's important to think about the nucleus and nuclear structure in 3D, and in fact, the heterochromatin domain, and in this case we're looking at a Drosophila cell, forms a distinct nuclear domain in 3 dimensions in interphase nuclei, and this can be seen by, on the right at the bottom, a concentration of the K9 dimethyl and HP1A, and on the left it's also seen as what's known as a DAPI bright region, or a region of condensed chromatin that leads to higher intensity of DAPI. So why study the response of these heterochromatic domains, and specifically repetitive sequences contained within them, to damage? And the main reason is that many of these repeats, as shown here on the top, such as AATAT, are present in multiple copies on non-homologous chromosomes. And if you get a homologous recombination event between these repeats, you create dicentric and acentric chromosomes. And this kind of aneuploidy has significant impact on viability and health in the form of birth defects and cancer. And because so much of the genome contains these repeated DNAs, if this type of homologous recombination is not regulated, there could be a very high frequency of dicentric and acentric chromosomes. So we're trying to understand how DNA damage in heterochromatin is repaired in a way that avoids such genome instability, and what, if any, is the role of chromatin components. And I'll tell you how live imaging has helped us with that. And first, though, I have to give you a very brief introduction to double-strand break repair pathways. If you make a double-strand break by irradiation or recombinases or any other of many techniques, there are 2 main pathways, non-homologous end joining and homologous recombination, and we'll focus on homologous recombination because that's what we found to be relevant to repair of double-strand breaks in heterochromatin. And basically what you get is recruitment of checkpoint proteins like ATM, then resection, recruitment of other proteins like RPA, and then finally you get recruitment of Rad51, which forms filaments and is required to invade homologous double-stranded DNA donors. And I've just highlighted these 3 proteins because they'll appear in the movies and in the rest of the discussion. Early on, you get phosphorylation of H2A variants in flies. This is known as H2AV. The gamma is for the phosphorylation. Another protein I'll focus on is ATRIP, which is recruited to resected filaments, and then Rad51. Okay, so previous studies had suggested that heterochromatin, being a compact domain in the nucleus, might in fact be recalcitrant to the formation or processing of double-strand breaks. And these were studies that looked at what you see here in these images, which are radiation-induced foci. So what happens when you get a double-strand break is that these repair proteins are recruited and concentrated at sites of double-strand breaks. And so what you can see in the upper left picture is Drosophila tissue culture cells. We've also done this in imaginal discs after irradiation. And what you can see is that there are a bunch of these green dots. These are the phospho-H2AV foci. And next to it is the grayscale image. And what's circled in the blue dotted circle is the DAPI bright region. And what you can see is that, in fact, at 10 minutes after radiation, you have formation of phospho-H2V foci within the heterochromatin as well as the euchromatin. But by 60 minutes after irradiation, in fact, you do not find such repair foci within the heterochromatin domain. And at the bottom you see quantitation of the kinetics. This is time after irradiation on the x-axis, and the average number of foci observed per cell. And on the left graph, you can see what happens in euchromatin, which is a standard repair response, which is that immediately you get formation of high frequency of these foci, and then a slow decline indicative of ongoing repair. And then, however, in heterochromatin, what you can see for the phospho-H2AV is that there's a sharp peak at 10 minutes after radiation, and then they disappear from this domain. So if we look now at the Rad51 protein behavior, what you see in euchromatin is kinetics that are similar to what happens with the phospho-H2AV, but in the heterochromatin you rarely, if ever, see Rad51 foci within that domain. So if we go to the next slide, basically from a lot of work, we've discerned that within the heterochromatin domain, double-strand breaks are very quickly detected and resected, that early components involved in homologous recombination, such as A-trip, are quickly recruited to these sites. However, these foci disappear from the domain by about 20 to 40 minutes after radiation, and they're not associated with Rad51 foci, that is, proteins involved in late homologous recombination at any time point. And yet, we've shown genetically that the repair of double-strand breaks in the heterochromatin domain, in fact, does require Rad51 and Rad54. And if we just go to the next slide, I'll show you. So the working model that we pursued was that, in fact, early and late events in homologous recombination are spatially and temporally separated. That is, within the heterochromatin domain, you get resection and recruitment of A-trip, but that, in fact, you can't complete recombination repair until these double-strand breaks move outside the domain. And so we went on to look at whether or not the heterochromatin domain displayed dynamic behaviors. And first, if you can run the movie, what you're going to see is that on the left is these are cells that have GFP tag HP1A, this heterochromatin protein, so you're only seeing the 30% of the nucleus. That's the heterochromatin domain, and you have to imagine the rest of the euchromatin and the rest of the nucleus. And on the left, you can see that before HR, this domain is pretty compact, and after irradiation, what happens is that this domain expands dramatically, and it also, you see these little fingers of HP1 sticking out and coming back into the domain. And so we can go on to the next slide. And this just shows the quantitation of those live analysis. And basically, what we can see is that the volume of HP1A increases about 1.5-fold. It starts as soon as we can begin the live imaging, which is 5 minutes after irradiation. The peak is at 30 minutes, and then there's a partial contraction. And this expansion and dynamic behavior of the domain lasts about 5 hours. But what we also observed was a very surprising dynamic behavior, and that is when we started to look at the live behavior of repair foci, what we observed was that they, in fact, did relocalize outside the heterochromatin domain after radiation. If you can run this movie, what we're looking at is the green is A-trip, this protein that recognizes resected filaments in early homologous recombination event, and the red is HP1. And what you can see is that immediately after radiation, there are very bright A-trip spots within the domain of HP1, and then as time goes on, by about 20 to 40 minutes, they get chased outside the domain and are only present outside of the heterochromatin domain. And so the relocalization that we observe starts about 5 minutes after irradiation and is finished by about 40 or 50 minutes afterwards. And just quickly, live studies have shown also something that we could not have imagined from looking at fixed preparations, and that is that if you follow these A-trip foci, they actually split after relocalization outside the heterochromatin domain, and this suggests that, in fact, the initial foci that we're observing are clusters of two or more double-strand breaks. Okay, and then if we, in comparison, look at how Rad51 behaves in live studies, we see a very different behavior. And if we start out just looking at the slides, I'll summarize what's observed, and that is that Rad51 foci appear later than A-trip, so they don't appear until 20 minutes, whereas A-trip is there as soon as we can image at about 5 minutes. They're only on the surface of the HP1A domain or in the euchromatin, and they also appear at these edges of these dynamic HP1A protrusions. So if you can run the movie, what you'll see is the HP1 is in red and Rad51 is in green. And in the upper left is a little cutout looking at one of these fingers that retracts into the main body of the domain and goes out again. So what you can see is that these foci are out at the periphery of the domain or outside the domain at all times, but also they seem to have an association with the domain as well as it moves in and out through these fingers. Okay, now we can do quantitative analysis of the relocalization of foci, and that's shown here. For the sake of time, I'm not going to go into detail, but you can compare the A-trip behavior. These are stills from these movies at the top with the different time points. At the bottom is a quantitation where we've looked at the solid red line is foci that are within the HP1 domain. Dashed line is at the periphery but still inside the domain. At the periphery or outside is a black line, and if they're outside, you see a green line. And we've done a kinetic analysis of this, and the main thing to focus on here is if you look at the red line, on the left, you see this peak of A-trip in the heterochromatin domain early after radiation at 10 minutes, and then it goes down immediately, whereas RAD51 is never there. Now, just in terms of approaches to how you do this quantitation, in fact, when we did these studies, we found that using automated segmentation methods was really not robust enough. This is a very dynamic domain, and you're trying to track these foci along with it. So it turned out that Rene Chiolo, the postdoc who led this study, had to do this through manual quantitation. You can imagine that's a lot of work. But we're now encouraged that we can do Pearson correlations to look at co-localization, for example, for A-trip and HP1, and we find that this is effective, an effective way to do automated quantitation of these types of movies. Okay, and then finally, the last few slides, we've been able to use live studies and looked at what kinds of proteins and mechanisms regulate these dynamic responses of heterochromatin damage. And what you see on the left here, repair proteins that we have identified as playing a role are shown in yellow. Proteins involved in heterochromatin function and formation are shown in green. There's also a summary of what we've learned about who's required for heterochromatin localization. For example, HP1A is required to recruit the SMC56 complex to heterochromatin, and what proteins physically interact by co-IP or mass spec analysis. And so the question is, which of these proteins affect relocalization and which of these proteins affect heterochromatin expansion? And basically, we've used the quantitative analyses. We deplete proteins by RNAi, and then we analyze the behavior of HP1 in terms of expansion and foci in terms of relocalization. And just very quickly, if you look at the top, we're looking at an experiment where we've depleted 3 proteins involved in resection. These are exonucleases. And what you can see in red is that if you, the red dots indicate the RNAi for the resection proteins. If you remove resection proteins, then you don't see any expansion of the heterochromatin domain. And also, if you look on the right at the top, here we're looking at, again, a kinetic analysis, but looking at foci located within the heterochromatin domain. And you can see that when you remove resection proteins, they no longer disappear from the heterochromatin domain, and they stay inside of that. That's the red line on the top. And so we've shown that these experiments show that resection proteins are required for both heterochromatin expansion and relocalization of foci. And if you look at the bottom, we've done similar studies looking at heterochromatin proteins, and these are experiments where you remove the SMC56 heterochromatin component. And what you find is that you don't have an effect on the expansion, but you do require heterochromatin proteins for the relocalization. And very importantly, if you look at the bottom right, now if you get rid of heterochromatin proteins, you can get RAD51 foci within the heterochromatin domain. Okay, and so just a summary of the analysis of regulation. What we've shown is that 2 proteins involved in late homologous recombination, RAD51 and RAD54, are in fact required for relocalization. And this suggests that there in fact may be a tethering event where foci that do relocalize outside the heterochromatin domain get attached, for example, to something that's located on the nuclear membrane, and that tethers them outside the heterochromatin domain. We've also shown that checkpoint proteins affect both expansion and relocalization, and then in terms of heterochromatin proteins, most importantly, HP1A is required to recruit SMC56, and it's SMC56, either directly or indirectly, that is responsible for blocking formation of RAD51 foci within the heterochromatin domain, and that this is in turn required for the double-strand breaks to relocalize outside the domain. Okay, so finally, in terms of relevance to genome stability, in fact, if you get rid of heterochromatin proteins, such as SMC56, then what you get are these massive aberrant homologous recombination events, and you can see these as fibers that connect cells that have failed to complete cytokinesis. And these fibers are covered with K9 methylation, indicating that they're heterochromatic. And so without the SMC56 complex, you are no longer able to exclude the formation of Rad51 foci, and you get massive aberrant recombination events. And so since my time is going quickly, I won't show the movie for the model, but basically, and you can look that up with a recent paper, supplementary information. And so basically what we've observed using live studies is that after irradiation, you get very quick processing of double-strand breaks within the heterochromatin. You get resection, recruitment of proteins that recognize resection filaments, but very quickly you get HP1A expansion of the domain. But completion of homologous recombination, that is recruitment of Rad51 and strand invasion, is blocked by the action of the heterochromatin proteins. However, what then happens is that between 20 to 40 minutes after radiation, you get movement of these double-strand breaks to outside the heterochromatin domain, and it's at this point that they've lost this block to Rad51 recruitment, and they can complete homologous recombination. So, and just finally, why are these dynamics and mechanisms important? Well, what these studies show is that the spatial and temporal separation of the early step, resection and strand invasion, the late step, reduces the probability of exchange between the double-strand break and undamaged repeats, homologous repeats concentrated within the domain, and that this reduces the probability of interchromosomal translocations and aneuploidy. So, and when these mechanisms are impaired, you get aberrant recombination and genome instability. And so, finally, let me just thank Evgenia Keolo is a fantastic postdoc who's done most of the work that I talked about, but she's been helped by Aki Minoda, Joel Swenson, Serafin Colmenares, and Sylvain Coste. And that's it.
Okay, thanks, Gary.
Sure.
Our third speaker is Dr. Jennifer Lippincott-Schwartz, who tell us about applying photoactivatable proteins and super-resolution imaging to the mechanics of cell migration and their properties of organelles.
Yes, thanks. So I'm going to continue the discussion of use of fluorescent imaging approaches for navigating the cellular landscape. Now, one of the things that is particularly exciting about this time in our sort of history of imaging is that these fluorescent probes, in particular these fluorescent proteins, are being used in very exciting synergistic applications that involve chemists that are developing the probes, biologists that are employing them in various techniques, physicists who are ever more developing microscopy advances, and finally the mathematicians that can take the quantitative data obtained with all of these approaches to really test mechanistic models for how particular subcellular phenomenon are behaving. Now, one of the probes that has been particularly fruitful in pushing imaging recently is the photoactivatable fluorescent proteins. These are proteins that become bright and visible upon being exposed to a pulse of UV light. And if you look in the bottom panel, you can see that pre-activation, the photoactivatable GFP, which we developed in my lab in 2002, can be switched on from a pre-activated dark state to a bright fluorescent state post-activation with just a very short pulse of UV light. Since the development of the photoactivatable GFP, there's been a whole cohort of photoactivatable fluorescent proteins that fall into three different classes. Those in class 1 that will photoactivate from a dark to a bright state can be either green or red. The second class, which can photo switch from one color to another. In the case of Kaede and EOS, they switch from green to red in response to UV light pulse. And then the third class of photoactivatable fluorescent proteins that can switch from being dark to on, either green or red, and then back off again by switching of 405 or 488 illumination. Now, what these photoactivatable fluorescent proteins allow researchers to do is to highlight selected subpopulations of proteins within cells. And if we move on to the next slide, which I'm trying to do, great. This is illustrated in the movie, which we can run right now, of a viral glycoprotein, VSVG, that's been attached to the photoactivatable GFP. And what you can see in the very beginning of this movie sequence, which if you can run it repeatedly would be ideal, is that we can selectively highlight the VSVG molecules that are traversing the secretory pathway at a particular place. In this case, we used the Golgi apparatus to switch on the molecules. And that then allows us to track the fate of these molecules as they leave Golgi and move to the plasma membrane without interference by other molecules that have not been switched on. Now, the use of these photoactivatable fluorescent proteins, as shown in the next slide, also permits the use in super-resolution imaging, in particular photoactivated localization microscopy called PALM, and its sister super-resolution single-molecule approach, STORM, which uses Psi3, Psi5 dyes that can photoactivate or photo-switch. To illustrate the power of PALM/STORM imaging, here's an aggregate of 50 nanometer polystyrene beads that have been coated with photoactivatable fluorescent protein Chi-8A. On the left, you can see the conventional image, which gives only an XY of about 200 nanometer resolution. And by comparison, if we shift over to the PALM image, you can see a resolution of 20 nanometers. And if we zoom up on that PALM image, as shown in this slide here, you can see we have the capability of seeing individual 50 nanometer beads within that large aggregate. Now, what I want to do in my short presentation is to just take you through some examples where use of these photoactivatable fluorescent proteins have been very helpful in not only addressing subcellular dynamic questions at the diffraction-limited imaging level, but also at the super-resolution imaging. So in terms of diffraction-limited imaging, one very powerful accomplishment that these photoactivatable fluorescent proteins can provide is to look at protein turnover. And the example that I'm illustrating in this slide is the turnover of a ER-resident protein called CD3 delta that's been tagged with a photoactivatable GFP. This protein localizes to the ER, which shown in the left-hand colored image is a fine reticular network that stretches throughout the whole cell. Now, if we photoactivate cells that are expressing the CD3 delta with a photoactivatable GFP at a time 0, as shown in the time sequence starting at 0, one can switch on all the molecules that reside in the ER at that particular time, and then you can follow the fate of those molecules over the succeeding time periods out to 6 hours. And what you can see is that these molecules are turning over very quickly, and that's because these molecules undergo a ER-associated degradation process that has been of considerable interest to people who are interested in how proteins are controlled at the level of ER dynamics. And what this is illustrating, if you look at the bottom panel where we have a percentage fluorescence over time, is the capability of really getting hundreds of data points to very carefully analyze the kinetics of turnover of this particular protein, but it could be done for any protein within the cell, and allow individual cells to be monitored and entire populations of proteins within those cells to be followed over time to get insight into key control points in the turnover of these proteins. Now, at the same time, one can monitor organelle turnover using photoactivatable fluorescent proteins. And this slide here just illustrates the use of this for monitoring the turnover of autophagosomes, which is shown in this cartoon up in the upper left-hand panel, represent organelles that form in response to stress or starvation and engulf portions of the cytoplasm that then are enclosed in a membrane bilayer that fuses with the lysosome. So one of the things people have been interested in the field is what's the lifetime of these organelles once they've formed. And we can do that by photoactivating a molecule that labels the autophagosome, LC3, and watching the lifetime of the fluorescence associated with that LC3 because it's photoactivatably tagged. And when you photoactivate that pool, that's the only molecule that is fluorescent within the cell. And you can track those molecules without any type of interference by other molecules that might become synthesized during the period that you're looking at. So if we look at the movie, which shows a pulse photoactivation of a population of autophagosomes that have been activated as a result of starvation, what one can see is that with a half-life of about 18 minutes, these autophagosomes undergo a process where they fuse with lysosomes and become destroyed. So that's just an example of how one can look at organelle turnover using these photoactivatable fluorescent proteins. But moving on to the next slide, I want to illustrate another type of application, diffraction limited, using these photoactivatable fluorescent proteins, and that is in looking at actin dynamics at the cell cortex. And if we could play the movie associated with this slide, one can see that the leading edge of a cell, in this case this is a slowly migrating cell, the leading edge is incredibly dynamic. What you're looking at is an actin that's been tagged with MRFP, and you can see that there's a peripheral pool of actin at the edge that's undergoing the dynamism, and then there's a pool of actin in these fibers in the rear of the cell. Now, if we go on to the next slide, what we were interested in testing using the photoactivatable protein TDEOS, which switches from green to red upon UV illumination, was to determine whether by photoactivating the portion of the actin cytoskeleton that's right adjacent to the plasma membrane, which is called the lamellipodium, converting selectively that pool from red to green by photoactivation, what is the fate of those molecules over time? In particular, we were interested in addressing whether these converted filaments are turned over by just a dynamic actin depolymerization pathway, or whether they actually are precursors for the formation of the actin arc-like structures in the lamella, where the focal adhesions are being laid down and play a critical role in the way that the cell crawls. And so the next slide shows essentially with the experiment, we're going to ask whether the actin filaments that we photoactivate in the lamellipodia are going to transfer into the lamella over time. And so in this slide that is shown here, what we're going to be doing is photoactivating way up in the lamellipodial region and then watching what happens to that converted pool. And if we show the movie associated with this slide, you can see that the red photoconverted actin molecules are completely turning over. So we see none of them being transferred into the lamellar region where the actin bundled filaments are localized. Now, if we do the same experiment, but now under a slightly different condition, and that is when the cell edge is actually undergoing retraction rather than protrusion, which was shown in the previous experiment. Now if we photoconvert the lamellipodial pool of actin and watch what happens over time, what one can see is that after the edge retraction, the photoactivated actin eos in red, shown in the right-hand two panels, is transferred down into the lamellar arc bundles. And this is illustrated in terms of a diagram in the slide that's shown here, where we think that what our data is suggesting is that the two pools of actin in this leading edge that undergo this dynamism that I showed in the first movie are actually spatially, temporally related to each other during the protrusion-retraction cycle of the leading edge, such that during protrusion, as the lamellipodial actin branched filament grows, there's no communication between that system and the lamellar system. Essentially, all the actin is drawn into building this network that's pushing out the edge. But once the edge reaches as far as it's going to extend and begins to retract, the actin now converts into a arc. We've shown that's a result of myosin 2 activity, and that sweeps this network down to form a bundled actin meshwork that creates the lamella. And so in this way, we think the two pools of actin at the leading edge are very intimately, spatially, temporally connected with each other. And the photoactivatable fluorescent protein technology was critical for really demonstrating this. Now, I want to now move on to how the photoactivatable fluorescent proteins have been useful for super-resolution imaging. Now, if we think in terms of a single fluorescent protein, which is about 3 nanometers in diameter, if one visualizes this fluorescent protein under a microscope, you're not going to see a 3 nanometer spot. You're going to see something much larger, about 100 times larger, and in appearance as a blurry spot, which is a result of the diffraction limit of the microscope. But because the individual proteins will give off a roughly Gaussian blurry spot, you can fit the Gaussian, the centroid of that Gaussian, with very high precision, and from that determine with very good accuracy where that molecule must be. The challenge, however, is isolating those molecules so that you can switch them on individually so that they're not overlapping. And that's what the technique of photoactivated localization as well as STORM is able to accomplish by sequentially photoconverting or photoswitching either the photoactivatable fluorescent proteins in POM or the Cy3, Cy5 dyes in STORM on from the dark state and fitting in different time frames the distribution of these molecules. Now, in the first set of experiments that I'm going to be showing you, we've done this, we've used this single molecule localization approach in fixed cells to look at the distribution of molecules associated with particular organelles. So here is an example of a reticulon protein, reticulon 4, tagged with the photoswitchable protein tandem dimer EOS, and we're looking three-dimensionally at the endoplasmic reticulum with very high resolution. Very beautiful capability of this strategy. Now, you can take these images and correlate with them with transmission EM, as shown in this example here, where we're looking at a mitochondrial matrix targeted switchable protein, and if we superimpose the POM image with a transmission EM, you can see very nicely the mitochondrial marker is distributing where it should be in these mitochondrial matrix region, which we think provides a really amazing alternative to immunogold labeling for high-density labeling of samples. Now, it's possible to do multicolor labeling with these approaches. Here's an example of two-color POM using a photoactivatable M-cherry and photoactivatable GFP that switch from dark to either red or green. And we're looking in this example at the nanometric distribution of transferrin receptor and clathrin-coated pits on the plasma membrane of cells. It's also possible to use POM in live cells. And if we show this movie here, what you're looking at is activation of these individual actin molecules. And if we move on to the next slide, we can fit the trajectories of these actin eos molecules, showing how they're moving with time. We can then plot the flow rates of these molecules to get velocity diagrams, and from that get further, deeper insight into actin dynamics at the leading edge of cells that are crawling like the one illustrated here. I want to just sum up by saying that using both the diffraction limited as well as the super resolution imaging approaches with these new fluorescent photoactivatable, photoswitchable proteins as well as dyes is really opening up a whole slew of possibilities for live cell single molecule tracking as well as super resolution ultrastructural analysis. And I want to end by thanking some of the key people involved in this work: George Patterson, who developed the photoactivatable GFP in my lab, and Harold Hess and Eric Betsick, who worked with us in developing the PALM and are now continuing their work as physicists at Janelia Farms. Thank you very much.
Thank you, Jennifer. And thanks to all of our speakers for their very exciting presentations. We're now going to take a few minutes to go over some of the questions that our listeners have submitted. But first, I'll get the ball rolling by asking each of the presenters one question, and that is, what do you see as the biggest challenge in your field in the ability to visualize cellular processes? Maybe we should start with Tobias first.
Yeah, I think in cell signaling, probably the biggest challenge is at this point not necessarily the spatial resolution, but it's when you look at GFP tagged signaling proteins. They're often present in very low copy numbers. So it's kind of a sensitivity issue. Raising the background of cells is fairly high when you image. So actually visualizing very low abundant proteins at physiological levels like growth factor receptors is probably one of the currently biggest challenges, to dig deeper into what most or all of the signaling proteins are doing spatially in cells.
And what about you, Gary?
Well, actually, ours is probably a more trivial issue, but actually, the issue that we have is how to image immediately after, do live imaging immediately after radiation. So the reason why we've up till now had a 3-minute lag, or at least the first time we could image expansion and relocalization was at 3 minutes to 5 minutes, is because Avine had to run from the irradiator to the microscope, and that was basically how much time it took. But now Sylvain Coste has actually set up a system where we can deliver X-rays on the microscope, and so the ability to immediately see the impact of whatever treatment it is you're doing seems to be is something that has, I think, held us back a little bit, but can now be fixed.
Thank you. And Jennifer, what do you think is the biggest challenge for you?
So one challenge is similar to Tobias's point, which is just getting molecules that are at low abundance labeled so you can visualize them without overexpressing them and potentially perturbing their behavior. But a second point is getting fluorescent proteins that are bright enough so that you can accurately detect them. And furthermore, approaches that allow us to count molecules in a reliable way. A lot of questions related to single molecule imaging that I think are very exciting relate to stoichiometry, and we need ways to determine how we can count these molecules in a reliable way.
So there are a lot of questions from the audience, a lot of interest, Jennifer, in your use of these photoactivatable proteins, particularly as applied to looking at degradation, and this may apply a little bit to the point you were previously making, but a lot of the folks in the audience were wondering how you do separate the effects of degradation from some other thing that might be happening with the fluorescent proteins, such as photobleaching or another reason that they might be going dark. And related to that, one one question was, can you discuss the relative advantages of photoactivatable fluorescent proteins versus just the regular fluorescent proteins?
Yes. Okay, so the turnover question is easily addressed by just doing a control where there's two controls you can do in the experiment that I showed with the turnover of the ER-associated protein. One is do the same experiment but fix your cells, photoconvert, and you'll see no turnover if you use the exact same imaging parameters if there is no effect of the fluorescent, of the light source in terms of bleaching. And we're using conditions where we have very little bleaching over the time course that we're collecting our images. So we've done that control, and it's not a problem. The other control that one can do is to inhibit the mechanism of degradation. And in the case of the protein we were looking at, the proteasome is involved in its degradation, and if we inhibit the proteasome, we see no loss of fluorescence over the time period that we're imaging. Again, supporting the idea that the technique is a viable one. But I should add that I think one needs to, if you're employing this kind of technology, you need to have the appropriate controls because, yes, you could easily get fooled if you did not do the appropriate controls because with too much light, these proteins will bleach, and you can lose fluorescence from that, from that type of process. And you need to distinguish that from the biological turnover. Now, related to just conventional fluorescent proteins, the challenge, we can't use the conventional fluorescent proteins to look at protein turnover because the conventional fluorescent proteins are being synthesized in steady state with their degradation. So you're continuously seeing molecules are continuously being synthesized at a comparable rate as they're being degraded. And so if you just image this ER-associated protein over time, you'll see no change in total fluorescence because molecules are being made and they're being degraded. And so you can't do this type of experiment with just regular fluorescent proteins.
Great. Thanks, Jennifer. We also had a couple of questions from the audience for Tobias, and those questions regarding about STIM1 translocation and how do you see that PKC is involved, and also a question about the cytoskeleton. How might the cytoskeleton be involved in STIM1 translocation?
Yeah, maybe I would start with the easier one first, which is about the cytoskeleton and STIM. So it has been shown by Abnova's group that STIM, so they did a screen for the proteins that interact with the plus end of microtubules and found STIM is a plus end microtubule binding protein. So it has a couple of characteristic sequences for that, and indeed, when you look along the ER, STIM proteins are moving in unstimulated cells across the ER with the help of microtubule polymerization. So this can change in some cells the distribution of STIM. However, when STIM is activated, it appears that this connection to the plus end of microtubules is inhibited. So it's a competition between being inactive and plus end bound, or active and free of the plus end. And we think the translocation following that to the ER plasma membrane junction is by a passive diffusion mechanism rather than by transport. The actin role in this STIM-ORI interaction is a bit less clear, but there's definitely actin there at the junction, and the role of that is as of yet not known. As to the PKC role in this STIM-ORI interaction, that's a very good question as well. So protein kinase C is one of these multifunctional protein kinases, and many, if not most, regulatory proteins have some phosphorylation sites that might be potentially phosphorylated by PKC, and in many cases have been shown to be phosphorylated by PKC. So in the case of STIM and ORI, there is as of yet no direct evidence whether these sites really have a significant regulatory impact, but PKC can affect many other important proteins in calcium signaling, so it's often difficult to to separate these effects that PKC has on a particular proteins from the effect it might have on the rest of the signaling system.
And Gary, there's a question for you, and this may be a little bit related to the question about probing how STIM1 translocates. Is you mentioned a potential tethering model for how heterochromatin or breaks within heterochromatin get translocated out of that region. And could you describe any approaches you're thinking about for testing that?
Yeah, so actually, I mean, especially in light of the other two talks, one important question is how does this relocalization occur? And, we can think of it as sort of active or passive. And we have removed microtubules, and we don't see any impact on relocalization. We're not sure about nuclear actin. But what we favor is the idea that this expansion of heterochromatin allows for more random movement of the resected double-strand breaks, and then but they basically get captured when they go outside the domain. And so the kinds of approaches that we're using is to get rid of proteins that are potential tethering proteins that are associated with nuclear membrane, and then do the live studies to see if they affect relocalization.
Thank you. And since we're out of time, we must come to the end of the session. And so we want to reiterate that if you missed anything during this webinar or would like to listen to it again, a recording will be available shortly on the CellPress website. And we'd like to say thanks again to all three of our speakers, Tobias Meyer, Gary Karpen, and Jennifer Lippincott-Schwartz, for their great presentations and for their thoughtful answers to the question session. And we are very grateful to our sponsor, BitPlane Scientific Software, for their contributions that made this webinar possible, and to Bright Talk for hosting it.
And we'd also like to thank all of you for tuning in today to learn more about the visualization and analysis of intracellular dynamics. Keep open your eyes and ears for more CellPress webinars on cutting-edge topics in biomedical research. And if you have any comments about this webinar or suggestions for future topics, we would love to hear from you. You can email us at cpwebinars@cell.com. I'm Karen Carniel.
And I'm Allison Evans.
And you've been listening to a CellPress webinar. Thanks for joining us.